Hello everybody. I'm going to spend the next 10 minutes on sensing methodologies and algorithms associated with smart agriculture and phenotyping. The first thing I would like to do is to introduce myself and my organization. I'm a research scientist in the Volcani Center, which is one of the largest and oldest agricultural research centers in Israel, which deals only with research and innovation in agriculture. We have 6 institutes which deal with all aspects of agriculture, from plant sciences, soil and water, plant protection, animal sciences, post-harvest, and ag engineering, which is the institute that I belong to. We have about 200 scientists and institutes from the center of Israel, which is about here, to the north and to the south. Specifically in deep technologies, we work on many aspects. Here I will concentrate on precision agriculture, where we have many activities in remote sensing, in sensing with different sensors, algorithms and computer controlled systems, as well as things that there are in agricultural disciplines, which, for example, irrigation and also food. I will start with some examples on data collection, things that we do, everything in research with collaboration with scientists in our institute and internationally. For irrigation and water management, we use the leaf temperature, which expresses the stress of the plants as an indicator of water uptake and water stress. We use the far infrared band from 8 to 12 microns in order to sense the energy which is emitted by the leaves. And we are trying to correlate it with agricultural parameters that they are used conventionally today to say something about irrigation, like the ones mentioned here. We use platforms like UAVs. In this slide, you see a helicopter, which is powered by fuel. It's a Spanish company helicopter. Unmanned Systems is the company, and it can be 2 hours taking images and scouting, having a payload of more than 2 kilograms. Using these technologies, we can see the temperature of the leaves, like we can see here in this slide. We can very easily distinguish between plants that they evapotranspirate, the low temperatures, the colors in purple, and high temperatures, and those that they do not evapotranspirate. We can very easily also see individual plants that they have excess water, like here. You can see individual drippers that they have excess water applied to the field, and we can just spot them. Using these very qualitative things, we can go ahead and process this information, and we can make decisions like where we have irrigation malfunctions. And we have found that the irrigation leaks, we can spot them or detect them quite accurately in a number of crops and in several times during the season. But maybe this is the easiest thing. The more difficult thing is to evaluate the water stress quantitatively and the irrigation scheduling. So we want to use the canopy temperature and find a way to correlate it with those agricultural parameters that are used for scheduling irrigation. In order to use that, to do this, temperature is not enough. We need an index or a normalization procedure that takes into account all the environmental conditions like air temperature, radiation, wind speed, relative humidity. And by doing that, we can build models that link between the agricultural parameter, which is in the y-axis here, and the thermal index, which is in the x-axis here. So we can translate any temperature on a given map to a leaf water potential or an agricultural parameter. And we can map this way the whole fields. This is about 20 hectares fields. We can map each one of the points according to its agricultural parameter, not just temperature. We can find the variability. We can make decisions by looking at the mean or variable irrigation by looking at the variability as well. We have gone one step further. We have seen that also the yield is the same, and the water use efficiency by using this remote thermal system is better than using conventional ways of measuring the water stress. But this is until now the challenge is to enhance this aerial imaging capacity to make it commercial. So one of the things that we are doing is led by lab in our institute, tries to use non-radiometrically corrected cameras and find algorithms to make the data that we have from those sensors reliable for sensing the water stress, and also using low resolution sensors in the thermal range in order to and use super resolution algorithms based on deep learning in order to enhance the resolution and make this visible in the large scale. Another approach is to use ancillary data, which means not just thermal imaging, but also spectral imaging, and combine them. And again, an effort led by Dr. Iftach Klap again is to develop imaging apparatus that are very low cost hyperspectral imaging systems that can be combined in those thermal images in order to enhance the performance. All this can be also applied in phenotyping. It's a big consortium led by commercial companies, tech companies, and agro companies in Israel that combine several modes of sensing, multispectral cameras, thermal cameras, and 3D sensing in order to define what is the status of the plant in terms of physiology, in terms of yield, in terms of biotic and abiotic stresses. We do it also in the field. An example of that is in the range of biotic stresses in orchards, where we have found that thermal imaging can detect the red palm weevil damage. And just an example is a map that we can produce here from thermal imaging that can detect with an accuracy of 75% potential trees that they are infected by this pest. Also, soil-borne diseases like in Fusarium, in Jopa. Again, aerial thermal imaging can be used in order to find the trees, measure the temperature, and we found there is a pretty good correlation between the current temperature and the infection level. They also can be used to find potential trees. All this is good in the field, but also in the greenhouses. Dr. Avital Behar leads a group that uses this kind of sophisticated sensors to monitor disease in the greenhouses. In this case, robotic arms go to the point where are the most relevant for making decisions about those diseases, and then the information that we get is more relevant about analyzing and making decisions about where the disease is. We also have some work on automatic traps where we use image processing and deep learning in order to detect flies which are caught in non-specific traps like this one. Images are transmitted to the cloud and they are set in order to identify the images. And last but not least, in the sensing techniques is the nanotechnology today, where we have a big effort in two directions. One is in non-structured porous biosensors, which is led by Georgi Sternberg. And you can see here there is a big number of applications that are very relevant of using those sensors based on silicon, also in the animal field and also in the water quality. And another approach is using bioelectronic and bioelectrochemical sensors, a lab that is led by Dr. Sam. And this technique is also in the nanotechnology, but uses different principles in order to identify even single molecules, aromatic molecules, or also they are used on biomass. So this is a very large and broad area that we work on the field of deep technology, photonics, and agriculture. Thank you.